Dear students, now we are going to discuss cofactors of enzymes. So what is an enzyme generally? Enzyme is generally a protein. So what is cofactor? Cofactors of enzymes. Co means accompany, in association with that. So here enzyme is with are made up of one or many polypeptides. So each enzyme is constructed with or made up of either one polypeptide or many polypeptides also, one too many. So basically, chemically, protein enzyme is a protein. So here, what about the number of proteins involved in the construction of one enzyme? One to many. One or two or three or many are involved in the formation of a particular enzyme. So in most of the enzymes, what are the chemical components? Actually, this heading comes under chemistry of enzymes. What about the chemical nature of enzymes? Chemistry of enzymes. Chemically, what are the enzymes? Chemically, what is an enzyme? Okay, so on the basis of chemical components present in enzymes, we divide enzymes into two categories. So on the basis of chemical nature of enzyme, what are they? So on the basis of chemistry, chemically, we can classify enzymes into two categories, two types. What are they? Okay, so first one, simple enzymes. Simple enzymes. Simple enzyme means enzyme with protein part only. So simple enzymes are with a protein only. Protein part only. Protein part only. Protein part only means they are without cofactor. These are the very simple enzymes. Example, pepsin. Generally, pepsin is a simple enzyme. Cofactors are not required, but it was not given in your textbook. Leave it, no problem. And the second one, second type, conjugate enzyme, holo enzyme, holo enzyme, or conjugate or compound enzyme. This is also not given in your textbook, but it is very, very important for NEET from here onwards. Holo enzyme, compound enzyme. What is this holo enzyme or compound enzyme? This enzyme is with usually two kinds of parts. What are they? What are the two types of parts? What about simple enzyme? Simple enzyme is with only protein part. Cofactors are absent. And what about a holo enzyme or compound enzyme? So holo enzyme is with two parts, majorly two kinds of part. What are they? First one, protein part. This is dummy diagram. So this one is protein part, organic protein part. So this protein part is called as apo enzyme. Apo enzyme. So apo enzyme is a protein part. And the second part is non-protein part. Non-protein part. That is not protein, other than protein. This non-protein part is called as cofactor. In your textbook, they explained from cofactors onwards. They didn't explain this part. Okay. So What is holoenzyme conjugate compound? Compound means more than one or not? Protein part, non-protein part. Protein part, apoenzyme. Non-protein part, cofactor. Now, what is the role of cofactor? Most of the enzymes are holoenzymes, compound enzymes, conjugate enzymes. For the catalytic activity of apoenzyme, cofactor is required. Cofactor is able to regulate the catalytic activity of apoenzyme. So the catalytic activity of apoenzyme depends on its association with a cofactor. So how many kinds of cofactors are there? According to a textbook, three kinds of cofactors. What are they? So cofactors are of three kinds. Okay, so what are they? First one, prosthetic group. Prosthetic group. Okay, so give an example. First example, heme. Heme is prosthetic group. PG, prosthetic group of two enzymes. What are they? Catalase, catalase, and peroxidase. 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 Okay, so heme is prosthetic group. And what is the role of these two enzymes? They are able to cause oxidation of hydrogen peroxide. These two enzymes concerned with hydrogen peroxide oxidation into H2O plus oxygen. So hyper concentration of hydrogen peroxide is toxic. That the toxicity is removed by catalases and peroxidase. So what about heme? So here heme is a organic cofactor. Organic cofactor. So this one is a heme, means a prosthetic group. Prosthetic group is permanently associated with a apoenzyme, not transient. Okay, so heme is a prosthetic group, one type of prosthetic group that is permanently in association with a apoenzyme part of catalase and peroxidase. So this association is not transient, permanent. Not only during the reaction, even without reaction also, heme is permanent part of catalase and peroxidase. So permanently attached. This is very, very important. So prosthetic group is differing from other cofactors, being it is permanently attached with apoenzyme part of those enzymes, permanently in association with that. What is that permanent? Even in the absence of concerned reaction, there is no concerned reaction, but it is part of that enzyme only. So there is no dissociation of prosthetic group from the concerned enzyme permanently, always, even in the absence of concerned reaction, which is regulated by that enzyme. Heme is permanently, prosthetic group is permanent part of that enzyme. So that is about a prosthetic group. Next. Second one, very, very important, coenzyme. Coenzymes. They are organic cofactors. Organic cofactor. This is one type of organic cofactor. So how many kinds of organic cofactors? Prosthetic and coenzyme. What are the examples? 1. NAD plus and NADP plus nicotinamide adenine dinucleotide. First one. Same, but phosphate is present. Nicotinamide adenine dinucleotide phosphate. Okay, so these coenzymes, NAD, NADP, generally they are concerned with they are generally concerned with redox reactions, reduction and oxidation. Go back to first class, oxidoreductases, malate plus NAD plus, malate plus NAD plus. Their enzyme is malate dehydrogenase. Malate dehydrogenase. Okay, so what about the cofactor of that enzyme? NAD plus. First reaction, go back to first reaction. Oxidoreductases. What about the given reaction? Malate plus NAD plus gives rise oxaloacetic acid plus NADH plus H. It was given there. Okay, so here I'm writing that enzyme also. Enzyme name is malate dehydrogenase with NAD plus as the coenzyme or cofactor. So what is the role of NAD, NADP? They are the electron carriers. Actually, electron carriers. We will study that one under the heading photosynthesis and respiration. Go back to 10th class. What about the end product of light reaction? ATP plus NADPHH plus. Okay, so NAD and NADP plus are electron carriers. They are able to transport electrons. Protons also correct. Both electron and proton carriers. So we'll see the function of these two at the time of photosynthesis and respiration. And these coenzymes, the association of a coenzyme plus apoenzyme. The association of coenzyme with apoenzyme is transient, temporary only. Transient, temporary, not permanent. Only during reaction, at the time of reaction only. Once reaction is completed, they dissociate from each other. So coenzymes are making association with the apoenzyme part at the time of reaction only. After completion of reaction, they move from that area to other area where they are required. And chemically, so-called NAD and NADP plus, chemically they are chemical components, vitamins. These two are with niacin chemical components. Simply precursor for the synthesis of NAD, NADP, vitamins. Okay, so that's about coenzymes, organic cofactors. Next one, last one, third one, inorganic cofactors. Go back to mineral nutrition, inorganic cofactors. Inorganic nutrients, magnesium, calcium, magnesium, manganese, zinc, acting as molybdenum also, acting as cofactors or not. Okay, so inorganic cofactors. So in this case, apoenzyme plus inorganic nutrients, minerals I am taking, minerals. Okay, so this type of enzyme is metalloenzyme. metalloenzymes. Metalloenzyme means enzyme is with the metal as a cofactor. Metalloenzymes. Okay, so what is the role of a inorganic cofactor here? So like coenzymes, inorganic cofactors also associated with the protein part at the time of reaction only. So what about inorganic cofactors? Here I'm giving certain number of examples. Mg2 plus cofactor of two types of enzymes. One is rubisco and pepcase. Go back to mineral nutrition. Okay, rubisco, ribulose 1,5 bisphosphate carboxylase oxygenase. Rubisco. We studied this one in biomolecules also. Go back to proteins. Pepcase. Next, Zn2 plus. Concerned with a cofactor of a carboxy peptidase. Okay, next. Molybdenum is cofactor of nitrogenase. And nitrite and nitrate reductases. Like that, we discussed so many examples in the case of mineral nutrition. What about the role of inorganic cofactors? For example, this is a enzyme. This part is very, very important. This is an enzyme, some enzyme, whatever the enzyme. Okay. So this one is substrate. This one is substrate, dummy one. This is substrate. Yes, I'm taking substrate. For example, take a zinc, Zn, Zn, and take certain number of Zns. So they are able to form coordination bonds. Coordination bonds between binding site and with the substrate also. One or many. Here one or many. Coordination bonds. Coordination bonds. Zn, coordination bonds. Okay, so for example, that inorganic nutrient is able to form coordination bonds with binding site of that enzyme. At the same time, coordination bonds, one or many with the substrate. What is the role of a so-called inorganic nutrient? Binding of substrate to the binding site. Fixing the substrate in the binding site. Okay, so that is about metalloenzymes. Go back to mineral nutrition. Nitrogenase. Chemically, what is nitrogenase? Molybdenum iron protein. So nitrogenase also conjugate enzyme, holoenzyme. So in nitrogenase, inorganic components, what are they? Molybdenum iron. Both. Molybdenum, given one, molybdenum iron protein. Protein is apoenzyme. And what about molybdenum and iron cofactor, inorganic cofactor? So that is a metalloenzyme. Okay, metalloenzyme. So that is about various kinds of cofactors which are able to regulate catalytic activity of an enzyme. If cofactor is removed, what about the catalytic activity of that enzyme? Declines or maybe absent. That cofactor is mandatory to regulate catalytic activity of that enzyme.